Ja, einen wunderschönen guten Nachmittag, meine sehr verehrten Damen und Herren. Der Vortrag war in Deutsch angekündigt. Can everybody understand German? Okay, so I try to do it in English. So today I'm going to talk about food fraud and countermeasures. So what can be done against food fraud? What we are doing food authentication. So this is a simply question of does it actually contain what it says on the package? So is the food authentic? So is it original or is it just fake? So in most cases it's a matter of profit why people are faking food. In some cases it can also be a problem, a health problem. But we are talking today about just about the food fraud. So what things can be faked? So there are different process parameters which can be affected. So production methods, so is the food organic produced or conventional produced? Has crop protections, chemicals been used, or fertilizer, synthetic fertilizer, and so on and so forth. Then the second question is about the geographical origin. So was the food maybe from South America, or from Europe, or from Germany, or from France? So these are the questions which have been addressed there. And the third point is the biological and also the chemical identity. So the biological identity, what species has been used or what variety has been used. And then the fourth point, this is also very important, and it's going directly into the field of food safety. Have been ingredients used which are hazardous to health, for example. So are there residues, contaminants, pathogens, allergens, and so on and so forth. So how can you protect yourself from food fraud? Just by authentication, so verification of the claimed properties. This can be in a first level, it can be done on the basis of documents. The problem is, as you can easily see here, we start normally, if you have plant-based materials, plant-based food, you start at the point of raw material cultivation. Then you have the harvest, the transport, the processing, the storage, distribution, packaging, and the wholesale or the retail at the end of the chain. So you have many players there, and as you know, documents can easily be created or modified by the user. You just need graphic program and a computer. So the next level could be visual or sensory inspection, but this is you need experts for that. So this is limited in resolution, and on the right side you see two different coffee species, Robusta and Arabica, or is it Liberica, or is it Excelsa? So it's very hard to distinguish just by eye between the species. So at the end, this is a very limited resolution. Another point or another level of protection are traceability systems, so digital traceability systems. So this starts by a one-dimensional barcode, goes over to a two-dimensional QR code, RFIDs, for example, you can use, so radio frequency identification chips on the package, but also blockchain technology is one of a technology which is used to protect yourself from food fraud. So the problem is that if you use the blockchain technology, the information is secure. If the information is in your chain, it's secure, but you always have the interface between the real world and the digital world. So at one point, let's say on the field, you get the information by the farmer, and then you put the information into the blockchain, and there the information is secure. But you have to make sure that the information at the beginning is reliable and was right. So this is also, I think this is a very good system, but you have to make sure that all the information you put into the chain is really reliable. And then the last point, and this is my point, is the experimental authentication. So it's completely independent of external information. It could be rather expensive, but it's very reliable, and it could be used at any point of the value chain. So, experimental authentication in routine. So you have a green and a red apple. So one or more markers are obvious or are known, and if you want to distinguish between the red and the green one, you just develop a method which analyzes the red dye or the green dye or some other points which are obvious. So this is clearly hypothesis-driven. That means that you know what you are analyzing. This could also be quantitative. This kind of analysis is called food targeting. But when you look on the process quality in this box here, you just can detect a few points out of your process quality question. So you can say something about the biological identity, so species might be different, and therefore you could easily develop a targeted method to distinguish between species. And if you know what residues are expected or what contaminants are expected, you can easily develop a targeting method. This is the challenge. You have two red apples, and you have no obvious differences which you can use to develop a targeted method. So in these cases, you have to look for differences. So here you use the molecular fingerprinting. So this is from the philosophy rather different to food targeting, because in this case you start just collecting data. So you have different technologies and you collect just data points. And for this kind of analysis, you don't need to know much about the chemical behind. So because you are just collecting data points, so it's called hypothesis-free method or a non-targeted method. And you need to collect as many as possible data points. So you have to be very high resolved. So you need very expensive technology. And you collect from your sample data points, but you can't say what data point is responsible for species A or species B. So in this case, you need the reference material to compare your data profile with a data profile of reference. So, and this could be very laborious because you have to collect the reference material all over the world. So if you want to develop a method to distinguish between, let's say, different geographical origins or between different provenances, you need the reference material from each location on the world which you want to use in your differentiation. Yeah, fingerprinting data collection, you can use a horizontal analyte spectrum, so beginning with DNA, going over peptides, proteins, small molecules, and last but not least, elements. So the technologies you can use are called the omics technologies. So from DNA, so you so in 20 years ago, one just analyzed a gene sequence or a gene segment. Nowadays, we are analyzing the whole genome. So the technology derived from this term is called the genomics. You also analyze the whole proteome, so the technologies are called the proteomics, and the whole metabolites, so the whole bunch of small molecules, this is called the metabolomics. And the elements, because you not just analyze elements, but also isotopes, is called the isotopolomics. So you have a horizontal analyte spectrum, and you have horizontal technologies which can be used for the analysis. So this is just horizontal. So the genomics means that you have different vertical technologies. So when you use the genomics, you can do in silico analysis, you can do NGS, so next generation sequencing, and microsatellite analysis, and so on and so forth. So if you carry out metabolomics, so for metabolomics, so for the small molecules, you can use NMR spectroscopy, you can use LCMS, you can use GCMS, so different vertical technologies which describe a part of the whole metabolome in this case. So the analytes which we want to analyze during the food fingerprinting, if you look on the genome, you have, when you use a plant genome, you have at least 300 million base pairs. When you look on the protein level, you have 30,000 different proteins you can analyze. In the metabolites, you have over 25,000 different chemical structures. And if you look on the elements and the isotopes, you have almost the entire periodic table. So what is the advantage of using different vertical technologies? I showed this point at the level of metabolomics. So the box here below shows that the entire box shows 25,000 molecules. We are at the moment, with the technologies available using LCMS, we are able to detect 5 to 10% of the 25,000 different molecules. If we use NMR, we are able to detect around 5%. If we use GCMS, we can detect also 5 to 10%. So, and what you can also see at this picture, that any technology addresses a different analytical window. So the molecules which we can analyze via GCMS are different compared to the molecules which we are analyzing using the LCMS. And they are also different with the group of NMR detected molecules. So what you can see here is just a finger, and this is the part we detect using the metabolomics. So we detect just a small part of the finger. This is a highly resolved fingerprint part, but not the whole finger. So, and if we use another technology, for example, isotopolomics, we analyze another part of the finger, also with high resolution. And you can easily imagine, if we use both technologies in parallel, the fingerprint gets better and better. And the best case would be to use all technologies in parallel. But you can easily imagine that this would be very, very expensive, and you also need the manpower to do this, you need the expertise to do this. And, but this would be in theory the best strategy. So we are, as I said before, we are collecting, doing fingerprinting, lots of data. And for that, to handle this data, we need chemometrics and bioinformatics. So we need databases, we need algorithms for data processing, and we need also algorithm for data, for merging the data sets. As I said before, when we use different omics technologies in parallel, it makes no sense to handle those data sets separately, but it would be much better and also much quicker to handle it together. And therefore, we need new algorithms. So in my group, there are informatics, so specialists working from data science, developing such algorithms. So, fingerprinting for real life. So the general problem of high-resolution fingerprinting is that you have this limited view on your finger. So if you just use the metabolomics, you see just a part of the finger. You need sophisticated scientific infrastructure, so you need very expensive instruments. You need high-resolution instruments. And you need scientifically trained staff. And this is all very time-consuming. You need also special laboratory infrastructure. So when we are carrying out DNA analysis, we are working in a clean room. So you need also the environment around your instruments. So in conclusion, this is all very expensive and also time-consuming, and it must become more cost-effective and also faster. So, and we have developed different levels of simplification, and one level of simplification could be low-resolution fingerprinting. So this is also a hypothesis-free technology. When you compare the fingerprint here with the small parts, with the low-resolution fingerprint, you see that you get much less data points, but over the whole finger. This is the advantage of this strategy. So this is also hypothesis-free. It's possible with relatively inexpensive instruments. So we are working with FT-NIR. So these instruments cost around 30,000 euro. If you buy a LCMS, high-resolution LCMS, you are over 300,000 euro. So it's much cheaper. So it's cost-efficient, and it's also suitable for routine use. And it's also easily applicable by trained technical personnel. And this is just an example, a pilot study where we use the NER technology, so the low-resolution fingerprinting. We developed a method to detect the shell content in cocoa products. So this is always a problem in chocolate fabrication. You start with cocoa beans, and normally the cocoa beans has a shell, and the shell has to be removed, because the shell is very difficult, because on the shell, if you have microorganisms in your or on your cocoa bean, they are located on the surface, so it could also be fungi. So if you have fungi on your shell, if you have fungi in your product, you have the risk that you get mycotoxin in your product. That means that you want to get the shell, the cocoa shell, out of your product. And therefore, we have developed a method. We started with LCMS, so with the most expensive technology, and then we developed a NIR method and compared this, and it's much more cheaper, but it's in a comparable performance range. So the simplification level 2 is just to do mini profiling, mini fingerprinting. So with the high resolution fingerprinting, we get lots of data points, and the question was to distinguish between different origin, to distinguish between different production methods. We use reference material, we have unknown sample and compare those fingerprints. And if the sample is not identical to the reference, we will see differences. And this you can see here. We compare the sample fingerprint with the reference fingerprint, and we saw a few differences. These are the red asterisks. And the simplification level 2 means that we develop on a second level a method which just detects these red arrows. And what you get as results, you can here you see the provenance Germany, Poland, Peru, and you see if you this would be around 25 or something like that, different molecules which we use for profiling, for mini profiling, and you can easily see that the provenances, Germany, Poland, Peru, and Netherlands are very different. So this is much more cost efficient. So the instruments you use here, you also work with LCMS instruments, are cheaper. And those instruments are suitable for routine use and could also be used by technical personnel. The next level of simplification is just single marker testing. So from the mini fingerprints, so from the differences between the reference and the sample, we just use one of these asterisks and develop a so-called lateral flow assay. I'm sure that everybody of you knows the lateral flow assay from the COVID-19 testing. So this is the same system behind. So it's a paper-based test system working with antibodies which are able to detect one single structure. What you get on the window of your test system are, in the positive case, 2 bands. One should be in any case there. This is the control band. And the second band, the test base, just occur if the test is positive. So if you detect your molecule from this mini fingerprint here, you get the second band on your test system. So this is very cost efficient. And it's also qualified for routine use, and it's a very simple application, and it's also possible for the consumer. So this is intuitively being used, and we are working on such testing systems on different foods. So all what I told you now was involved in a big project. It ran over 5 years. It was called the food profiling project. So we have there different groups working with high-tech method development, so the non-targeted or the fingerprinting, and also the development of targeted application for routine work, and also bioinformatics, chemometrics have been there, and one of the main points of this project was also technology transfer. Yeah, that's it. And last but not least, I want to thank my group and also my cooperation partners, also the money provider, and last but not least, I want to thank you for your attention. Thank you.